So welcome everyone to the third seminar of the 2016 seminar series organized by STAPA. So STAPA is the Young Researchers Association of Institut Pasteur, and we organize different activities, social activities like the famous Beer Hour, also many activities to integrate the incomers, the new people, especially students. But we also organize some scientific events like the Young Researchers in Life Sciences conference, the Forum Biotechno, this series of seminars. So this series of seminars are, the idea is to have some seminars that are not about very specific data, so that are more like wide for all the community in Pasteur. Before we had the first seminar was given by Stephen Friend, who is the president of Sage Bionetworks. He told us about how to integrate the community into biomedical research by using, for example, applications in smartphones to do clinical trials and diagnostics. Second was Thomas Pradou from the University of Bordeaux, who works in philosophy of science, specifically in philosophy of biology and in theoretical immunology. He discussed with us about the role of, or the impact of the concept of microbiota and the construction of symbiont relationships, and how this challenges the concept of biological individuality and the development of the understanding of the development of organisms, and also some concepts about the immune system, not only as an army that defends us against pathogens, also as a homeostatic, its homeostatic role. And now it's time, thanks to, actually have to mention that Media Richetti, what is Media? Thanks a lot, because she was very kind in contacting us and telling us that Antonio would be in Paris on June. So she encouraged to contact him and ask him to give a seminar. Well, Antonio is a very known scientist in the field of origin of life. He has been working in this field during 35 years, and he has been visiting scientists in different universities in Alicante, in Madrid, in Switzerland, ETH Institute, in Russia, in California, and also here in Institut Pasteur, right? So he has devoted a lot in education and in communication of science to general public. He has written the book The Origin of Life, which is a bestseller, and he has been also member of the board of the Society of the Study of the Origin of Life of the NASA. He was actually the president of this society for 2 terms, being the first Latin American scientist to occupy this position. So he will tell us about the biochemical roots of the RNA world hypothesis, and I'm very thankful. Thanks a lot for being here, and we look forward to your talk.
Thank you very much. The first thing that I would really like to say is that I'm very grateful to Pedro Hernandez Cerda and all his fellows and friends and colleagues for the invitation to be here. I'm extremely happy to be here. In Mexico, we say that the babies are brought by the cigon, by the storks from Paris to Mexico. So it's a very well-rooted tradition. So I think it reflects, at least in part, this strong attachment that the Mexican and Latin American culture has to the French culture. And being here for me is really being very much at home. So what I would like to do today is to speak a little bit about the history, the actual history of how the RNA world concept developed. RNA went from being the ugly duckling of molecular biology to becoming a very central actor or actress, if you wish to be politically correct, in the scenarios describing the origins of life. And I will also touch a little bit on the likelihood, or perhaps the unlikelihood, of forming all the components of RNA in the so-called primitive soup. So, having said that, let me start by trying to put the things in context. We have witnessed a major intellectual, conceptual revolution in biology when we became aware that there were many different ways in which we could describe the history, the phylogeny of living entities. But of course, when the use of ribosomal RNA and other macromolecular markers led to the phylogenetic tree, we all agree that the traditional Aristotelic or Linnaean or Whittaker views of life dividing into two main kingdoms or three main kingdoms or five kingdoms was fine at a certain level, but if we look at the molecular markers, then we can clearly see the existence of three major domains. Now, we can reconstruct without any major problem, so to say, the traits of the less common ancestor, or the less universal common ancestor, as some people like to call it, just by comparing, making a statistical description of the traits that are found in all the tips of the branches of this universal tree. And this will lead us to the qualitative description of what appears to have been already a very complex population of microbes engaged in lateral gene transfer and so on. But when we speak about the origins of life, we are actually addressing an issue that's really further down in the basement of this building. When we speak about the origins of life, one should not forget that if not in time, at least in complexity, the first living entities were in fact very different from the less common ancestors. So we have to address an issue in which we need to explain how metabolic pathways appear, how the genetic code developed, how membranes first evolved, and so on. And this is something that we have to keep in mind. Now, so far, for all the criticisms that have been raised against the theory, what really forms the basic scheme for trying to understand the origins of life was the book that was published in 1936 in Russian, in 1938 in English by Alexander Ivanovich Oparin. You could call him a biochemist, plant physiologist. There are really no contemporary equivalents to what he studied there. And by assuming a very Darwinian attitude, Oparin came to the conclusion that there was no such thing as the spontaneous generation of life. Something that in this place had already been shown to be the case. And Oparin actually developed a proposal that is multi and interdisciplinary by assuming that instead of a sudden emergence of life, what we had was a stepwise evolution that started with the appearance of organic compounds in a reducing atmosphere. He addressed the idea, he accepted the idea of a reducing atmosphere by talking to astronomers, by talking to planetologists, something that was very unusual at the time for someone coming from the life sciences, something that is still very unusual for many life scientists. And by relying on the fantastic, extraordinary German tradition of organic chemistry, he was able to propose that in the primitive waters of the environment, of the primitive Earth, there was the synthesis of organic compounds. Eventually we learned to call this the primitive soup. And as he was always very much against the idea of assuming that there is one such thing as a living molecule, he assumed that conglomerates of the molecules formed under the primitive Earth had led to system that he thought would be best modeled by quasorbates. We don't speak of quasorbates anymore, but we just think of them as historical curiosities. But at the time, they were the best model, the best physical chemical model for what the people call protoplasm, which is also a term that we have forgotten basically, except for the historians of sciences. And from there, he assumed that the first metabolic pathways evolved, which were basically based on glycolysis. After we had fermentative anaerobic glycolysis as basic primordial pathway. Some of you may wonder why Oparin did not include in his scheme nucleic acids, but don't forget that the book was published in 1938 in English, and at the time, you can check it on the web, James Watson was in 5th grade of grammar school. So clearly the people have no idea of the significance of what nucleic acids meant. But the theory was so open in the epistemological sense that it could easily adapt itself and accurate understanding as the description of biological phenomena proceeded. Well, nothing much really happened when the book was published in 1938 until 1953, because in 1938, of course, we already had the Second World War with the downfall of the Spanish Republic, and most of the scientists were actually engaged in one way or the other in war projects. There are fascinating descriptions of Max Perutz designing places where the airplanes could land, of Crick designing anti-mining devices and so on. So not really much happened in science, in the biological sciences, until after the war started. And then in 1953, a very young, unassuming chemist from the University of California started to do his PhD under the direction of Harold Urey at the University of Chicago. The young chemist was, of course, Stanley Miller, and after convincing his advisor that it was worth trying to simulate the conditions of the primitive Earth, he actually made up, set up an experimental design, which you can see in the left part of the screen. And after only 5 hours, he was able to show that you could accumulate a number of organic compounds without any problem in an atmosphere that had methane, ammonia, water, and hydrogen that was activated by electrical discharges. And unfortunately, Stanley Miller threw away the original apparatus. He threw it away because he told me several times it was very difficult to clean it, so he decided to do with it. So a very famous experiment ended up in the trash bin of the University of Chicago Department of Chemistry. But we can repeat the experiment without any problem. He left impeccable descriptions of how to do it. And indeed, you can see how only very few hours, you can see how there's a chemical change inside the apparatus. I like always to show these two photographs because it also demonstrates very clearly how the standards have changed in doing chemical experiments. I should say that the upper flask was covered with asbestos, with amiant, which is something that no one in his right head will use today. But it also shows how the dressing standards of the students have changed. The people used to go with white shirt, tie, cufflinks into the lab, and nowadays not even professors dress like that. But in any case, the experiment proved out to be quite a success. Stanley Miller did an analysis, a chemical analysis using paper chromatography, and he was able to show that it was very easy indeed to form aspartic acid, glycine, alpha-alanine, beta-alanine, which we rarely find in living systems, alpha-amino and butyric acid, and a few other amino acids. The paper made quite a splash when it was published, but Stanley kept the original samples that he had used, he had recovered from the experiment, and because I was lucky enough to be a very good friend of his, he gave me the original samples, and eventually together with my friend and colleague Jay Beda, who was a graduate student of Stanley Miller, we decided to undertake a very careful analysis, chemical analysis, using the available techniques today, which are of course extremely sophisticated. So when Stanley Miller reported only 5 amino acids, we actually revisited the experiment, and you can see the huge amount of different compounds that we were able to detect. Of course, we were able to corroborate his original results, but you can see the huge amount of different amino acids and amines that may actually be underrepresented here because they tend to evaporate. Everybody who has worked with amines knows that they very quickly evaporate. You can smell them in the lab, so we may be actually missing some of them. But what is fascinating is that, and it's a point that I would like to make, is that when we look at the outcome of the experiment, this is the original experiment, we repeated it as well, but you can see a number of protein amino acids, but also you can see non-protein amino acids. For instance, you can see valine, which is a protein amino acid, but you can also see that isovaline and norvaline were also formed. And if you actually look at the abundances, isovaline was much more abundant in the sample than valine. So I think this really shows that one shouldn't have the simplistic idea that one had the first cells emerging from the primitive soup. there was some sort of chemical selection, physical selection that may have to do with the stability of the compounds, with the nature of the bonds they form, and so on. But clearly this is showing that valine may be in contemporary proteins because it was available in the primitive earth, but we have to understand why this is not the case of isovaline or norvaline. One point that I really like to stress a lot is that we don't have a sample of the primitive earth. We don't have a sample of the primitive atmosphere. We don't have unmodified geological samples of the conditions of the earth when life appeared, but we have meteorites. Meteorites actually can have different ages. Here you see a picture of the Murchison meteorite that fell in Australia in September 1969. By then, NASA had all the infrastructure to analyze the lunar samples, so the meteorite was analyzed right away. It's an ongoing work, and here you can see the huge number of different compounds of biological significance. We see, for instance, that practically all the decarboxylic acid that we find in the Krebs cycles are there. We see about 90, in the Murchison meteorites, we see about 70 different amino acids. Most of them, not all of them, most of them in racemic mixtures. Again, an issue that this is the right place to speak about, and many other compounds. And the meteorites, the chemical analysis of meteorites validates in many ways the experiments we are doing. Let me show you an example of an experiment that, again, at the level of my friend Jeff Beda in UCSD in La Jolla was performed. We analyzed the aliphatic amino acids present in a sample of the Murchison meteorite. You see them in blue. And then we actually repeated the Miller experiment by adding hydrogen sulfide, and you see an impeccable correlation both in qualitative and quantitative terms. So this is telling us that indeed our experiments are a good simulation of the chemistry of the primitive solar system and suggest that indeed there was a primitive subform quite easily before life appeared. Now, one point that I really want to stress a lot is that we tend to forget that in 1953, not only the Miller experiment was published, but also that 2 weeks before it was published, actually the Watson and Crick model of the double helix was proposed. It was in April when Watson and Crick wrote in Nature their model, published in Nature their model. Stanley Miller published his paper on May 5th, which is, by the way, the day in which the Mexican army defeated completely the French army who was invading Mexico at the time. No hard feelings. But what is fascinating, what is absolutely fascinating, is that with the publication of the DNA double helix, all of a sudden, all biology changed. Actually, the impact that was made by the Miller experiment was much greater, but I think you will all agree with me that the publication of the double helix model represents a landmark in the way we perceive biology. All of a sudden, a number of fields in life sciences, like paleontology, or like the natural description of the insects and plants and so on, were displaced to a second-rate type of science. And in particular, if you wanted to have a reasonable theory of the origins of life, clearly it had to coincide, it had to follow what molecular biology was telling us. So clearly it was not enough to publish only the synthesis of organic compounds like amino acids. You see, it was required to have a connection between our models of the primitive Earth and what molecular biology was showing us to be the case at a very basic level of the structure and substructure of cells. And this is what makes so important the prebiotic synthesis of adenine that was achieved by my friend, my teacher, my colleague, Juan Noro in Houston, where he was working and could demonstrate without any problem that hydrogen cyanide that you see in the upper left part of this slide can condense quite easily to form a dimer, a trimer, then a tetramer, and with the presence of formamidine you can have the aminimidazole carbonitrile, which with the addition of formamide forms adenine. And then in 1966, Jim Ferris and Leslie Orgel took a small flask, some samples of aqueous solution of the amino trial, then, as we call it in Origins of Life, went out into the sun in the Salk Institute, and in a few minutes there was a photoisomerization took place, and you can see how actually again you can form the aminimidazole carbonyl trial without any problem, and then you go into adenine. Scientifically, this is impeccable. It's a beautiful reaction, but it also shows that there is indeed a connection between molecular biology, we are forming one of the components of nucleic acids, of ATP, and so on, and the origins of life field. So this sort of validated in the eyes of the origins of life, of the molecular biologists, the field of the origins of life. A few years after the experiment was done, we decided to repeat the analysis of this excited imidazole group with the aminocarbonyl trial imidazole in the middle, and we were able to show that very easily you could form not only guanine and adenine, but also the decomposition products, not because they were decomposing, but because they were forming at the same time. And later on, we decided to repeat the oral experiment, but looking now at the supernatant in the solution, and we simply took an ammonia derivative, heated up in a simulation of Darwin's one pond by drying and hydrating, and we were able to see very clearly the huge peak of adenine, which was not a surprise, a very huge peak of guanine, which was not a surprise either, but then because we had the standards, we could actually detect the presence of 2,6-deaminopurine, for which there is no report, as far as I know, of its actual presence in biology, except from a 1970 report of its presence in a virus reported in the Soviet Union. We're not quite sure that the standards that were available were not contaminated, so it's not clear that we can really find it in biology. But then we decided to play with the structure of the molecule, and let me just show you how 2,6-deaminopurine base pairs with uracil. And we therefore proposed that perhaps in the primitive Earth there was a wider range of bases available for making double helixes. In other words, that perhaps the number of bases available for primitive genetic polymers was greater than the 5 we see today. And very soon indeed, very recently, it was shown that there is a tremendous array of purines and pyrimidines that we can synthesize under prebiotic experiments, trying to simulate the Miller conditions, and also you find them in meteorites. So this is clearly showing that we are convinced of that both data from the analysis of meteorites and from the simulation experiments strongly suggest that there was no such thing as pure RNA or pure DNA or pure protein-like compounds in the primitive Earth, but the diversity, the chemical diversity was quite extraordinary. And again, we have to understand how this selection, what kind of processes took place that selected the bases and the amino acids that we see today from this very complex mixture. Now, everybody will agree in one way or the other that it's quite easy to have this so-called primitive soup. And the question, of course, is how do we get from there into DNA, RNA, and proteins? And I do not need to go into the details of the fascinating arguments and discussions that took place during the 50s, the 60s, the 70s, in which the people were saying that DNA came first, others were saying that no, it was protein that came first. It was a discussion that was actually tainted by political perspectives. Some of my most leftist friends prefer the idea that proteins came first, and when you ask them why, they will say, well, Engels says that protein, albumin, came before in the dialectics of nature. Many of them were not aware that Engels had actually taken the idea from Haeckel. It was a very popular idea at the time. And some of my most conservative colleagues actually say, no, DNA is the master molecule. And all kind of anthropocentric models developed in which DNA was called the marshal of the army of molecules inside the cells, or the manager of cells and so on. Well, obviously we know that what came before is DNA. And the question is, how did we come to this conclusion? And I think it's in itself a fascinating story. So let me start by saying that a broken watch, as the English will say, gives the right time now and then. And you can get to the right conclusion using the wrong premises. And in the 1950s, many people were aware, thanks among others to John D. Bernal, the professor of Crick, of Rosalind Franklin, and so on. They were very much aware that some viruses are indeed endowed with RNA as genetic material. It was a very clear-cut demonstration of the presence of RNA in viruses, the tobacco mosaic virus being the ultimate example. And at the same time, many people were assuming that because viruses are very simple, therefore there must be primitive. This is not necessarily correct. If you look at the case of intestinal parasites, it's clear that they are simple but not primitive. If you look at Ikebana, the wonderful Japanese arrangements, it's very sophisticated. It's not primitive at all. It's not a precursor to the very Baroque flower arrangements that the French or the Mexicans like. So clearly simplicity is by no means an indication of primitiveness. But at the time, the people were convinced that since there are viruses with RNA, and viruses are on the threshold, and perhaps some people would say represent the first living entities, therefore RNA genomes must be primitive. So it was the right conclusion based on wrong assumptions. But it was quite right. The other thing that happened at the same time is that in the 1950s, Brachet, who was in Belgium, and Belozersky, who was in the Soviet Union, independently came to realize the significance of RNA. The Camembert diagram that you see on the right part of the screen is of course based on modern data, but when you look at the original papers by Belozersky, by Brachet and others, you realize how very keen these people were on trying to look at the different components of living cells in terms of dry weight. And it was perfectly clear that protein is by far the most abundant component, that you had structural components like lipids, phospholipids, cofactors, and so on, and that there was a big slide, about 1/4 of the total weight of cells that corresponded to nucleic acids, and the bulk went to RNA. Belozersky, who in many ways can be considered the founder of Russian Soviet molecular biology, was actually able to show that when you have a bacterial population that's growing, you actually have an increase in the amount of RNA that's being produced. And Brachet, who was an embryologist in Belgium, was able to show that, in fact, when you have an embryo from a zygote that's developing, which can be seen as a population of cells that is growing, the same phenomenon was observed. Brachet, at the time, was a member of the Belgian Communist Party. He had a good sense of eventually leaving it, so he could travel back and forth between Belgium and the Soviet Union a lot, and there was an extraordinarily fruitful amount of discussion between him and Belozersky. And this led to the recognition that indeed RNA could have appeared long before DNA. And there is a fascinating couple of paragraphs that Belozersky published in 1959 that he presented on the first International Meeting on the Origins of Life in Moscow in 1957. And he says, it's worth reading because he says, there is no doubt that nucleic acids play an important role in the evolution of the organic world and metabolic reactions. Yet, both RNA and DNA could hardly arise simultaneously in the early evolution of life. It rather seems that ribonucleotides and RNA originated first. DNA came into existence far more recently as the protoplasm became more differentiated and its functions grew in complexity. It seems that RNA, being associated with the most general processes of life, was formed at an earlier evolutionary stage, while the origin of DNA was associated with the development of more specialized and phylogenetically later features of organisms. So here you have someone that was a biochemist, but had a tremendous influence of Darwinian theory, and I think this represents what at the time was developing very beautifully, which was the field of evolutionary biochemistry. Eventually died, it basically died, but I think that these statements by Belozersky show how refined were the people's thoughts about the evolution of biochemical processes. At the same time, independently, quite independently, mostly in the United States. Philip Handler, who was a major figure in the USA science, he was actually president of the National Academy of Sciences, a very, very accomplished chemist, together with Eakin, who was working in Austin, Leslie Orgel, who was by then already in the United States, and Harold White, realized that if you want to have enzymes, most of the enzymes require cofactors, require coenzymes. And they actually did quantitative analysis to see which portion of those coenzymes were actually derivatives of ribonucleotides, and they realized that many of the most famous FAD, NAD, ATP, and so on cofactors are indeed ribonucleotide derivatives. So they came to the very bold conclusion independently of what Beloselsky and Brachet were saying, independently of each other, they came to the conclusion that perhaps it was an indication that in very early stages of evolution, metabolic pathways were actually catalyzed by the coenzymes, but not by the enzymes themselves. So the stage was set, in fact, to the point that from the early 1950s onwards, I think that it's fair to say that the role to proposals of the RNA world was played by the embracement of the idea that primitive life had RNA genomes. This was generally accepted by the people, by Haldane, Bernal, Pirillo, Parin, Beloselsky, Brachet, Lippmann of the ATP proposal, that the suggestion that ribonucleotidyl coenzymes were vestige of an ancestral metabolism, and then in the 1960s, 60s, the extraordinary awareness by Smithis, by Crick, by Orgel, by others, that in fact the very complicated three-dimensional structures that you can see in RNA and not in DNA could be an indication that perhaps RNA could have catalytic activities. And this was clearly demonstrated when the people realized that if you want to have protein synthesis, I like this figure, of course everybody knows it, but I think it's fascinating because it shows how you can have protein synthesis in the absence of DNA. And the people not only realized it, but also realized, as Francis Crick wrote down, that transfer RNA molecules, which by then had been already crystallized, actually have a very complex three-dimensional structure comparable to what we see in proteins, and therefore they might be an indication of their catalytic activity. So what you have here is an extraordinary insight of people that were very much aware of the developments in molecular biology. They knew their chemistry, not only the molecular biology, they knew their chemistry very, very well. They were developing the basic traits of what we later will call molecular biology, and this led in a natural way to the so-called RNA world hypothesis, the idea that in the primitive Earth, prior not only to DNA but also prior to proteins, RNA was encoding for catalytic RNAs that was helping to assisting the replication of RNA. And I think it's a fascinating hypothesis that was the outcome of biological intuition, an evolutionary framework, something that was lost eventually among many molecular biologists, and of course a very good understanding of molecular biology. There's an additional component that I find absolutely fascinating. The hypothesis of RNA coming before proteins and DNA was suggested absolutely independently, completely independently, by Alex Rich in 1962, by Carl Woese in 1967, by Francis Crick in 1968, and by Leslie Orgel in 1968. I strongly recommend you to read especially the 1968 paper by Francis Crick published in the Journal of Molecular Biology, because that by Leslie Orgel, because they are masterpieces of intuition and evolutionary analysis. But there is an additional point that I found absolutely fascinating. I had the honor to be a very good friend of Leslie Orgel. Through him I got to know Crick. I could see how they were always exchanging ideas and so on. And then I decided to go into the archives, which are available at the University of California in San Diego, and it's fascinating to see how the people were willing to exchange very freely their hypotheses. It was a very small group that saw each other as a colleague and not as a competitor for grants. So I think it's very sad to see how science has developed nowadays, in which people are not that willing to expose to others their wild hypotheses or their very deep analysis, because they're afraid of having them stolen before they are published. At the time, no. At the time, the community was very, very small, and the atmosphere of free discussion is absolutely superb, both at the personal level or at the level of letters of, that were being exchanged. I think you can actually see them now in the web. Well, so the RNA world hypothesis was published, was proposed, and I think it's fair to say that during the 20 years that followed the 1968 publication of the papers by Leslie Orgel and Francis Crick, everybody, basically everybody, accepted the idea that RNA genomes in cells came prior to DNA cell genomes, that many people accepted the idea that coenzymes were vestigious evolutionary remains of an earlier stage in which metabolism was catalyzed by RNA, but nobody was really working on it. Some accepted the idea that histidine was a molecular vestige of an RNA world stage, and the only one that actually worked on it was my friend and colleague Marie-Christine Morel, who's sitting here, who, when she was working with Jacques Mignot, actually decided to develop this idea and actually demonstrated that histidine is a very good analog of adenine for a number of biochemical reactions. At the same time, there was the development of in vitro evolution of RNA systems by Sol Spiegelman. Actually, he came here to Paris, explained his ideas in the 60s, so that you had Carl Woese here as a postdoc and say, no, you have to come with me to the USA. So he stole Carl Woese from the Pasteur Institute. But at the same time, he was a very, very clever scientist that came to the idea of developing methods to use the Q-beta replicase and the Q-beta virus to actually demonstrate that you could evolve RNA molecules under laboratory conditions. But the main point is that nobody was really working on the possible catalytic roles of RNA. Nobody thought about the idea. Everybody thought it was just a wild hypothesis. I think that mostly because all of us have been educated with the idea that catalysis depends on proteins, and RNA was perhaps a curiosity, but nothing more than that. Well, you know that RNA catalysis was discovered basically by chance, independently, by the group of Tom Cech and by the group of Sidney Ullman. And there's a fascinating paragraph in a paper that Sidney Ullman and his colleagues wrote in 1983, and it says that if proteins were relative latecomers in the evolution of macromolecules, then primeval manipulations of nucleic acids may have been carried out entirely or predominantly by catalytic nucleic acids themselves. The remnants of these early important biological events may be apparent in the reaction of RNase-P, sorry for the misspelling, in the processing of some ribosomal RNAs and in ribosomes, and undoubtedly in other reactions catalyzed by RNA that remain to be identified. And I think all of you will agree with me that the extraordinary discovery that the peptidyl transferase reaction is catalyzed only by RNA in the large unit of the ribosome is a very clear-cut demonstration that indeed protein biosynthesis started in a time, in a stage in which RNA was already available in the primitive earth. So I think this is one of the best demonstrations that we have that indeed RNA came prior to proteins as we understand them today. So, what are the implications of the RNA world hypothesis? Well, if you look at the extant natural ribozymes, most of them are actually involved in RNA processing and in protein biosynthesis. And I think there's a mantra that we have to learn, that the ribosome is a ribosome. The ribosome is a ribosome. If you learn this, you understand the basics of the early evolution of protein biosynthesis. Secondly, clearly, both the catalytic and structural properties of the ribosome strongly suggest that protein synthesis evolved in an RNA world in which amino acids were already available. And this implies that the origin of the genetic code and the origins of life are no longer synonymous. I think this is important because for many, many years, several decades, many physicists were actually engaged in what I call Pythagorean mathematics, because they were trying to find solutions, some sense of cosmic harmony between triplets and amino acids, and so this has essentially failed. clearly we want to understand the origin of the code, we have to understand the biology of RNA molecules. And finally, I think it's fair to say that since DNA replication is catalyzed solely by proteins, a huge array of proteins, I don't need to describe them, many of you can do that much better than me. Clearly, DNA is a later outcome of evolution and not necessarily as RNA. Well, but there are an additional number of traits of RNA that all of you are aware of them, that I would just like to underline. RNA can be involved in cell regulation of its expression via the ribo switches, and if we look at RNA